Welcome to another deep dive. Today, we're exploring a really fascinating and highly specific set of source materials that take a hard investigative look at the clinical trial documentation behind childhood vaccines. We'll be relying entirely on this source data to unpack how safety baselines are actually established in modern medicine. That includes direct excerpts from FDA package inserts, historical documents, and legal analyses. So if you love getting into the nitty-gritty of scientific study design and historical data, you are absolutely in the right place. Okay, let's dive right into this. The central question driving our material today is simply this: how are childhood vaccines tested for safety before they're approved for public use? Now, it sounds like a totally straightforward question with a simple answer, right? But as you'll see, a deep dive into the documentation reveals a testing landscape that is way more complex than you might expect. Here's our roadmap for today. First, defining placebo control. Second, analyzing FDA package inserts. Third, the active control chain. Fourth, the 1986 liability turning point. And finally, analyzing the first vaccines. Section 1. Defining placebo control. Let's establish the scientific baseline required for standard medical testing. Okay, so before we can really look at the trials, we gotta get our definitions straight. According to the FDA's own guidance, a placebo is strictly defined as an inert substance with no pharmacologic activity. And the CDC echoes this, defining it as a substance or treatment that has no effect on living beings. Typically, in a gold standard clinical trial, an experimental group gets the new medical product, and a control group gets this truly inert placebo, like a simple saline injection. That is what allows scientists to isolate the exact effects and the true safety profile of the new product. But here is where things get really interesting. The documentation presents a pretty striking contrast. On one hand, you've got public claims from major medical institutions stating that childhood vaccines are carefully studied, including with placebos, to ensure safety. But on the other hand, the primary finding from the source material, after combing through the actual FDA licensure documents, is that not a single routine injected childhood vaccine on the CDC schedule was licensed based on a true inert placebo-controlled trial. Which brings us to Section 2, analyzing FDA package inserts. Let's look directly at the data manufacturers actually submitted to the FDA. When you dig right into Section 6.1 of the FDA package inserts, a surprising pattern pops up. Take the hepatitis B vaccines, specifically Recombavax HB and NGRX-B. The trials used absolutely no placebo control. For the DTaP vaccine, the control group didn't get saline, they actually received an older DTP vaccine. And for the pneumococcal vaccine, Prevnar 13, the control group was given an older version of the vaccine, Prevnar 7. So instead of measuring against a neutral inert baseline, these trials were measuring new vaccines against other active vaccines. And then there's the duration of the safety monitoring itself. Get this. The FDA documentation reveals that the Recombavax HB vaccine was licensed for babies based on a trial that monitored safety for just 5 days after injection. Another Hep B vaccine, NGRX-B, was monitored for just 4 days. The source material highlights that this window is just far too brief to detect any long-term neurodevelopmental or immunological issues that might pop up later. This practice leads us right into Section 3, the active control chain. Let's trace how this testing methodology actually compounds over time. It's kind of like a chain reaction, and the pneumococcal vaccines illustrate this brilliantly. Step 1. Prevnar 7 was licensed based on a trial where the control group received an investigational meningococcal vaccine, meaning they got an unapproved experimental vaccine, not a placebo. Step 2, when Prevnar 13 was developed, it was tested against Prevnar 7. Step 3, when PCV15 was developed, it was tested against Prevnar 13. The logical risk of doing it this way is captured perfectly in this quote from the source material. You're compounding the risk that something downstream has been done incorrectly. By never establishing a truly inert baseline of safety at the very beginning of the chain, it means that any adverse events caused by the earliest vaccine just become the new normal baseline. So if both the new vaccine and the active control cause serious adverse events at a similar rate, the FDA can deem the new vaccine safe because there's no numerical imbalance. And that's even if the absolute rate of adverse events is high. So you might be wondering, why are trials designed this way? That brings us to Section 4. The 1986 liability turning point. First, just consider the sheer scale of the change in the childhood schedule. Back in 1983, a child received about 3 routine injections in their first year of life. Today, following the full CDC schedule, a child can receive 25 or more injections before their first birthday. The source documentation argues this massive expansion was directly triggered by a massive shift in legal liability. That shift happened with the 1986 National Childhood Vaccine Injury Act. This law effectively granted liability immunity to vaccine manufacturers, meaning if a child is injured by a routine vaccine, the family generally cannot sue the pharmaceutical company. Instead, they have to go through a government compensation program. The source explains that removing financial liability completely inverted the economic incentives. For normal drugs, companies run long-term trials to ensure safety and avoid massive lawsuits. But with vaccines, manufacturers no longer have that same financial self-interest to conduct rigorous, long-term, true placebo-controlled trials because their liability risk has basically been removed by the government. Finally, Section 5, analyzing the first vaccines. Let's trace this methodology back to its roots. We're talking about one of the most famous medical trials in history, the 1954 Salk polio vaccine trial. Now, the popular claim out there is that the control group received a simple saline placebo, just salt water. But when you look at the official 1954 report, it tells a wildly different story. If you go right to page 51 of the final report, the control injection included a synthetic tissue culture medium known as 1999 solution, phenol red, antibiotics, and remarkably, formalin, which is a solution of formaldehyde. It was definitively not an inert saline placebo, which means even the foundational trial of modern vaccinology lacked a true biologically neutral baseline. Which leaves us with a really profound question. If foundational vaccine safety trials, from Salk's polio vaccine all the way to modern additions on the childhood schedule, lacked true inert placebos, how do we establish a true baseline for medical transparency and safety today? It is a super compelling look at study design, the actual definition of a placebo, and the data hiding right inside FDA package inserts that challenges us to look closely at the primary sources of medical history. Thanks so much for joining me on this explainer, and keep asking great questions about the data.